Hello, everyone. My name is Mike Temple. I'm the lead healthcare data scientist at TriNetX. I've been there for about 3 years. As far as background goes, I'm actually a physician by training. I practiced medicine for 15 years, and I've been doing data science work for the last 10 years. Um, I'll pass it over to Zuza for a brief introduction for her.
Hello, my name is Zuzanna Drebert. I'm also a data scientist at TriNetX. I've been working here for about 7 years, and I have experience working with healthcare data for about 8 years now.
And we're going to talk about our experience using unstructured data and from our network. So just a little bit about TriNetX. We are a provider of real-world data. We are a global, we have data from across the globe in Latin America, North America, Europe, and Asia Pacific. Our largest presence is in North America, but we do have a significant presence across the other areas as well. We partner with healthcare organizations in each of those regions. They provide us de-identified data from their for their electronic health record. We then use that data to provide back to the healthcare organizations to assist in their research efforts. We also work with life sciences company to help them find patients for clinical trials. So we do help with clinical trial recruitment in these various areas. So this is just an overview of kind of patient counts in across the globe. And this is a kind of another view of kind of the size of our network and how it's been growing over the past several years. Currently, we're over 220 million patients around the globe, and over 200 healthcare organizations globally as well. In addition to the structured data from the electronic health records, we also receive clinical notes, unstructured data from a portion of our healthcare organizations. So we have over 65 healthcare organizations in the U.S. 17 of those currently provide us clinical notes. That number is growing. We are growing the note network every year. From these 17 HCOs, we have over 350 million notes that we have access to. These notes are of various types. They include progress notes, admission and discharge summaries, radiology notes, surgical notes, procedure notes, laboratory reports, etc. So we do have a very diverse amount of a large number and a diverse amount of notes across our U.S. network. So the use case that we're going to talk about today is extraction of smoking status from our unstructured clinical notes. And why are we doing this? There are several problems with the structured electronic health record data. One, smoking is often incomplete or missing. There are diagnosis codes that capture tobacco use or nicotine dependence, but these studies have shown that these are poorly documented in the codes, and they're not often maintained. So if a patient's a smoker and then quits smoking, those codes aren't often updated. They're just carried over, and so it looks like the patient may continue to be a smoker. Obviously, smoking status is a very important data point for research, quality metrics, risk stratification. Obviously, there are a lot of high risk populations due to smoking, and so we want to be able to identify those smokers for research studies, for clinical trial inclusion, et cetera, for new drug development and new research purposes. So, and there have been some studies that have shown in the structured fields alone, almost 87% of smoking status is missing. By adding free text notes, we can reduce that number to below 30% of missingness. And some of the work that we've done at TriNetX, we can actually get that number below 30%. So what do we hope to capture? We're trying to capture actual status, so current, former, or never smokers. And then where available, we're hoping to capture the some sort of quantification of their smoking. So that may be packs per day, pack years is a common calculation. So any sort of quantitative measure that we can add to the patient's smoking status. So that is what we are trying to extract from the unstructured data that is not readily available in the structured data. So why is this difficult? Like I said, we have 17 HCOs currently providing us notes, and they all have a wide variety of documentation. So, for example, this is just some examples that we have in our notes. We're going to talk about these three HCOs in our talk today, but once again, this applies to all 17 HCOs. So, for example, HCO A, you can see examples in the top left. They have more of a section header approach. So they have like a social history and then a tobacco section and then like a tobacco use kind of topic tag. HCOB in the middle tends to use more of a bulleted approach, right? So they have a bullet list when it comes to documentation of their smoking status. And then HCOC at the very bottom, they have somewhat of this kind of section header or topic heading structure, but it's kind of punctuated with all these question marks. And so this makes it difficult for baseline models, NLP models, to chunk these out into sentences. You can see that the punctuation is missing or not consistent at all. So it becomes difficult to chunk these into sentences and then get them down into even smaller chunks. So we have to really create fine-tuned models for each healthcare organization. And Zuza will go through that process, but we use John Snow Lab, and we also do our annotations in GenAI Lab from John Snow Lab. And so because of this variety in documentation, we have to create annotation guidelines for each HCO. So 17 different HCOs and growing. So you can see here on the right side is an example of some of the annotation rules that we have. So for example, these are common phrases that are present in this particular HCO. And then this is our rules around how we would document the name entity recognition, how we would document assertion status, and the quantity fields for each of these examples. And we do this so we can get annotators to agree on the what should be documented when we're creating our gold standard notes to help us evaluate model performance and to do the fine tuning of the models. So we create rules for annotations for each of the HCOs as we go through, and then the annotators have those rules they can follow as we annotate these notes. I will go ahead and pass it over to Zuza to go through the process that we do.
Thank you, Mike. Yes, our annotation process is an iterative process to generate gold standard documents, which then can be used to fine-tune or train models that we then use to extract information from a large amount of data. First of all, we have to make sure we collect and prepare relevant notes. So in case of smoking status, we are looking only at progress notes, and we are also not taking full documents, which can be several pages long, but we only take fragments of the text to limit amount of data to be processed. We are also cleaning the data a little bit. For example, we are removing hidden Unicode sequences that can affect tokenization process. And we are also, depending on the source, cleaning the data so we can facilitate sentence detection, as Mike mentioned before, and this is very important for assertion prediction. Then when we have such prepared nodes, we apply them to our pre-annotation pipeline, where we are using pre-trained models from Snow Labs and NER, a deep learning model for entity recognition. And here is a smoking status, smoking type, substance quantity, frequency, and duration. And this is followed by a assertion model, which predicts assertion for smoking status. And then when we have documents pre-annotated with these models, we upload them to GenAI Lab annotation tool, where our human annotators review and correct the pre-annotation accordingly to our internal annotation guidelines. And when they are finished, we have first batch of gold standard documents ready. We split this data set 80-20 for training and testing, and we are using the training subset for fine-tuning the NER model and training the assertion model, and we evaluate performance on the test subset. When we have first models ready, we can then reuse them for the second batch of nodes. So we pre-annotate the second batch with these models, and then we pass it again to annotation tool, where these nodes, these pre-annotations are corrected, and we have second batch of gold standard documents that is used again for another round of fine-tuning and training. And we repeat this cycle multiple times until we obtained the performance. In our case, our goal is to obtain precision, macro precision of above 0.9 or higher. And when we have models ready, we can use them to further extract information from large free text data. Now I will go over some fine-tuning results from our 3 example HCOs to show you how this fine-tuning process goes. So as we mentioned, we are using Snow Labs tools and pre-trained models for this purpose. And here on the left, you can see the precision recall and F1 score for the original model versus fine-tuned models. So you can see after a few rounds of annotations, we have successfully fine-tuned the model to obtain precision of our goal. And if you look on the graph to the right, you can see each entities, each label separately. For example, smoking status is above 0, is 0.94, and other entities are also of high precision. I just wanted to mention here that the models that we are using as a base models, pre-trained models from the library, this model was much broader than just smoking status. For example, here, substance duration, frequency, and quantity. It was also detecting alcohol and other drugs, because that was the goal of that base model. And of course, the precision baseline precision was low here because our guidelines, our goal was only to target smoking-related entities. Next slide, please. And we are also training an assertion model. So for smoking status, we want to know whether the person was a present smoker, it was past history, it's absent, the status is possible or someone else. And in case of assertion model, there is no option to fine-tune an existing model. We have to train a model from the beginning. However, Snow Labs provide tools to do it, and with only few rounds of annotations, we managed to train a logistic regression model that is performing quite well already. Next slide, please. And these are the results for fine-tuning a NER model for the second hospital. You can observe here the results are very similar to the first model. So although the data formatting, the data source was much different here, after several rounds of annotations, we managed to successfully fine-tune the model to reach very high precision for almost all of the labels. The precision is above 0.9. Next slide. And similarly, we have used Snow Labs tools to train an assertion model, a logistic regression model. However, in case of this healthcare organization, it was more difficult to obtain macro precision of above 0.9. So we might still work further to improve performance for this healthcare organization. Next slide. And the third HCO, again, we have very good results for fine-tuning. We have obtained very high precision after a few rounds of annotations. All the entities, all the labels here have a very high precision. So another successful fine-tuning experiment. Next slide. And also in case of this hospital, we have trained an assertion model. We have reached high precision for almost all of the labels here, and the macro precision for this hospital was above 0.9. Next slide. As we mentioned earlier, the last step of our cycle is to reuse the models that we fine-tuned, that we trained, and extract information from large amount of data. And this is an example of how this data can be harmonized across different HCOs. We can provide data in a structured form containing such fields like NER chunk, which is the segment of the text from the document, chunk position, the NER label, so smoking type, smoking status, substance quantity, and so forth. The confidence of the prediction from NER model. And we have assertion label column that is only applicable for smoking status. And also we have assertion confidence score for this prediction. Next slide, please. We have been using Snow Lab tools for several months now, and we have learned a lot. We have very promising results, and we have also learned some challenges and limitations during this time that we would like to share. So first of all, probably obvious limitation is that correcting annotation is a time-consuming process. It requires skilled annotators and very good annotation guidelines to make sure the annotators can annotate in consistent way. This is certainly the slowest step in our annotation process, but we cannot avoid it. Each use case, whether it's a smoking status or biomarkers or medication details, will require own rules and guidelines. And a lot of research has to be done to analyze how this data is recorded and what is the best way to annotate it. We also observed diversity in how the free text is formatted across different institutions and different EHR systems. So we also have to change our annotation rules and modify them accordingly to different formatting styles, and we had to create separate models and separate gold standard documents for each HCO or group of HCOs. Of course, extracting data from nodes is compute intensive. As we mentioned, we have millions of nodes available in TriNetX, so processing this amount of data is very costly. So we also take measures to make sure we limit the amount of data that we process by pre-selecting relevant nodes only, and where applicable, to use only fragments of nodes instead of full documents. And the last challenge that is a goal of TriNetX, which is to harmonize the data among many healthcare providers. So regardless of the source, we want to make sure that the data can be queried and analyzed in the same way across different institutions. And this is also applicable to the data extracted from the free text. And this is all from us. Thank you so much for your attention.